I'm very happy to be here, and I appreciated the fact that ISMB was interested in getting into the synthetic biology area. And I think we've had already three very interesting talks. These talks were all composed independently, and I think at this point you will recognize that there are certain themes that keep coming back that are important in the execution of this whole field. One of them is the reuse of modules that can be connected in one way or another to make new materials. So what I'd like to do is to talk about those, what we call bricks, which are the elemental modules that get connected in order to achieve the kind of functionality that we need. So I'm going to talk about the motivation for doing it, creating new functionality, and in part this will be summarizing some of the work that's already been done and presented earlier today. I'd like to then try to go into the semantics and the actual ways of expressing what we know about these modules and compare the current efforts that people are doing in the ontology areas to things that in fact have been done in the past, and see if we can't avoid some of the unfortunate difficulties that we've had previously. Finally, I'd like to take you through what I think are some of the key issues going forward and indicate where you might be able to find some very recent references which suggest that a lot of these ontological problems have a good prognosis. So what we mean by the bricks is basically elements that we can chain together. And I've taken a little cartoon here that's asymmetric and just shown you that in fact in order to really make these things work together you have to have them organized in a manner that they fit and that in being used they don't interfere with each other. What it really means is that you have to have restriction sites upstream and downstream of each one of these individual parts, and the restriction sites have to achieve a functional connection one with the other, or this part simply isn't going to work. The particular sequences that I have up there here on the slide were ones that came out of one of the very early BioBrick definitions, but at this point, obviously, there are many different ways that you can do that in a creative fashion. But one of the important things is that it be documented how each one of these individual parts can attach to others to make a construct. If you put these together, you can achieve the kinds of and, or, nor, XOR types of functionality that have already been described. And I think, in fact, if you remember the one slide from Ron Weiss, one of the examples was creating a a signal at the center of a field of cells and having different gates go off such that when the signal was very high, which means that the signal's now diffusing out from the center of this circle, that when the signal was very high, you got a green fluorescence, and when the signal was very low, it was lower at an intermediate value, you got a red signal, and finally, when it was farther away, you got no signal at all. And this is a three-step function, but obviously you can get more and more. One of the most important driving features of the progress in this field over the last 5 years has been a program called IGEM. Is there anyone in the audience besides the speakers who have participated in IGEM in the past? Well, I see a surprisingly healthy show of hands. The IGEM program has been wonderful in the sense that it has energized many laboratories around the world, and it has grown at a compound rate of about 20 or 30% a year over the last 5 years. Last year there were well over 1,000 people, and this coming fall there are going to be 3 regional conferences with a final IGEM conference at MIT again, and all of the constructs that these teams develop all go into this open architecture where you can reuse many of the constructs. So between the impetus given by IGEM and serious money put in by organizations such as the EU and the Wellcome Trust, particularly the generous funding that the Wellcome Trust has given to Professor Kitney's lab, and to some new money that's coming out of the Defense Advanced Research Projects Agency in the U.S., I think we're going to see major advances in this field in the next few years. So let's look back now to think about how we can put a semantic wrapper on this field that will allow us to all talk to each other and to share a lot of the concepts that are being developed. It's useful to look back at similar kinds of situations where we had multiple points of light, multiple laboratories developing individual solutions, and then having a situation where everybody would like to use the commonality of those solutions and finding that there was a severe lack of ability to communicate. One that comes to mind that Professor Kitney and I worked on some time ago is the digital image communication in medicine standard that was originally started back in the mid-'80s. It was very unsuccessful. It was an attempt by the users to try to break the monopoly that Siemens and GE and Philips had on the equipment that was purchased, so that they didn't have to be slaves to the particular company that they bought the equipment from. It took all the way to 1993 before there was an international standard that was adopted. And that standard eventually, over the next 5 years, certainly by 1998, 1999, covered all of the individual information areas that you see here: patient monitoring, patient folders, diagnostic imaging, lab data, and the like. Now, one of the problems that came up was that this consensus on the particular standard for how all of this is recorded in, for example, diagnostic databases, all had to be hammered out in committees and agreed to before any kind of standard was published and used. So the vocabulary was totally controlled. The big difficulty with that is that takes a lot of time, and it becomes very politicized, and the amount of progress that you can make is very low. I'll give you just one example. What we tried to do in my lab around 1990-2000 was to say, we're going to take all the great things that DICOM did and helped establish for the medical imaging, and we're going to try to apply that to experimental biology. And let's take an example. For example, a microscope. Microscope puts out images. We should be able to reuse a lot of this information. We went ahead and made an information object definition in the same sense that it had been done with the medical imaging. And what you see on this diagram, all the little boxes here were basically large chunks of definitions about, in a microscope you have, let's say, an objective, you have filters, you have the particular details of the microscope and the camera and so forth and so on. And all of this information has to accompany the image in order to make it robust and useful. Well, what we found, unfortunately, was that after we had made this information object definition, we went to one of the laboratories over at Harvard Medical School and said, we can help you. We have this great technology. Well, it turns out that they had different definitions. And so we were at loggerheads. And the problem was that in order to make any further progress and to get their information out of their database and into ours and vice versa, we had to make this agreement with the semantics. So that was a real problem. And what we're trying to do, I think, and what I've perceived, particularly from the current literature, is that it's very possible to get by that step by using basically communication pipes between the different data sets. So the lessons that I would take from this is don't get trapped in semantic fights. Try to use synonyms and try to use information sources that are machine parsable. For example, RDF is one of the World Wide Web language standards which you can use to code up information from ontologies, and you can in fact use an RDF store and write a access routine that will go in and get the information you need out of that store without having to have a committee convene somewhere and bless the particular semantics that you're going to use in order to interpret the RDF store that you're getting data from. Now, that doesn't mean that you won't find it important to have minimum requirements for these data sources. There are a lot of acronyms that have come out of the Europe and the UK, and Miriam is a classic example. It's minimum information requested in the annotation of biochemical models. And each one of the constituents of a biochemical model can be annotated with known information from a lot of different resources. Here, for example, the model itself could have a digital object identifier. It could be listed in Medline. It could be listed in PubMed. The gene ontology also has many models listed. So if you say, I have a model, and it corresponds to something in the gene ontology, then there's additional information that people have that they can go to the gene ontology and extract that. If you have a reaction, for example, it may appear in some collection such as KEGG. And each one of these sources that you can put references to increases the amount of information that your particular construct and your particular data set can have. Friday and Saturday of this past week, there was a biology open source community meeting, and they talked in some detail about the semantic web open source issues. The basic result of that is that people are trying to go back and forth between different data repositories, basically by writing software bridges. Okay, let's talk about the future. It's very clear from the previous talks that we're moving into quantitative models. We need to be able to predict what these models are going to do even before we do the experiments. The number of different sources around the world where these models are available continues to increase. One of the most important things about these sources is the curation. And as you heard from Professor Kitney, in some uncurated collections, such as the BioBricks at MIT, there may be thousands of constructs, and yet it's much more difficult to create the kinds of curated lists of parts as they have at Imperial College. Nonetheless, the curation step is incredibly important because if you manage to properly and accurately characterize the parts that you have, they're much more useful than something where the behavior is unknown. Two efforts to describe, to define languages that are extensible and allow data collections to be parsed and extended. One is the synthetic biology open language. That's primarily being worked on at the University of Washington in Herb Saro's lab. Michael Golzinski is doing a PhD thesis on that. Imperial College is also heavily involved in that. There was another effort called Clotho, which came out of the University of California, Berkeley, and a portion of that effort went to Boston University with Douglas Densmore recently. Clotho is really the idea of trying to set up the equivalent of cloud computing so that you can have a site on the web where you can have functionality that you can address without having to know the details of how the functionality works. Finally, one of the efforts that has been around for some time is called BioPax. It had DARPA funding in the early 2000s, and it has recently been revitalized about a year ago. Peter Karp is giving a talk on some of that material at 3 o'clock this afternoon in this same room if you are interested in coming back. Let me just take 1 minute or so and give you an example, just in summary, of the steps that you of curation and description that you need to go through to get a well-characterized part in synthetic biology. This example comes out of NTU in Singapore. Anand Saidi is a PhD graduate student there. The basic idea is that you produce a sensing molecule. You're trying to see if the molecule in green the protein in green is there. This sensing molecule then combines with the one you're trying to detect. That then comes in, upregulates the production of GFP, and therefore the GFP signal is a function of the fact that the green molecule is present. So in this sense, this is a gated process, and you can actually look in this as a quantitative model that would evolve in time. You can go to Cell Designer, for example, and you can describe how it's set up. You can then annotate all of the species involved. For example, here there are like 9 species. The annotations come from databases like PubMed, UniProt, the Go ontology, and so then the temporal behavior of this system, starting from known initial conditions, is described by a series of ordinary differential equations. And you can see a number of the LASR and AHL molecules that you saw previously in some of the other constructs. So you can solve those equations, you can predict what's going to happen, and then you can do the experiments and find out whether the predictions, which are the dark lines, correspond to the actual experimental behavior, which are the dots here on the observations. So the question is, where do we go from here? I think that the databases and the functionality we're trying to do involves models, quantitative models, time-dependent models. It means lots and lots of characterizing experiments. It means comparisons between the models and the experiments, and as Professor Kitney said in the very beginning of his talk, that you have to feed the results of all of that back into the models and continue to iterate them and to expand them and to improve them. To the extent that we can share information with between these different applications, I think that the way to do that is to in fact be able to write access routines to address well-characterized open source databases like UniProt and Go, and then build your own applications to do that for your specific purposes. So I thank you very much. I hope that if we are able to bring back another synthetic biology program next year and the year after, that we'll see some of you at that program. Thank you very much.